Hello everyone, welcome to Lecture 2 of Biotechnology and Society. I initially thought of doing an actual lecture on Google Meet for this topic, but because of my experience yesterday and today in my classes, I opted to do this recorded lecture. And this was also the request of my students this morning because of technical difficulties we had, both on my side and on their side. That's why this is what you instead. Anyhow, we do what we have to do so that you will not be shortchanged when it comes to learning. So moving on, this was your second assignment, and I hope you enjoy this very short article. I also hope it gave you a lot of ideas for possible research and perhaps for future entrepreneurship ventures. So just imagine the many possibilities from this biotechnology, such as edible plastic packaging for noodles. I know that many of you love your ramen so much. So imagine throwing in a pan of boiling water your noodles plus its packaging. Now that would be definitely good for the environment. Let's have some bits of trivia first. Do you know this? If you're a pizza lover like me, then you recognize that it's a piece of cheese, but it's not an ordinary cheese. It is actually blue cheese. Now that is something interesting. So I checked the price of blue cheese last week in preparation for this class and found out that in the Philippines, for 1.5 kilograms of blue cheese, it would actually cost more than 4,000 pesos. Now, I'm sure that you are already thinking and asking, why is it so expensive? You will learn later on. So it is blue because it has the fungus Penicillium roqueforti added, and that's why you see blue spots and some kind of blue venation. Actually, we know that bacterial species are used to make cheese. So for blue cheese, different bacterial species are actually used. And then after the cheese is ready, it is added with spores of Penicillium roqueforti to grow on the cheese in order to achieve the blue venation. And actually it stinks, but it is very expensive cheese. And the reason for that is, accordingly, it has some medicinal properties and it actually reduces cardiovascular diseases. So that is the trivia for this lecture. Today we are going to focus on the history of biotechnology, although it would be impossible to present to you a complete rundown of its history. Nonetheless, I have titled this presentation with a bit of drama. So it says, revisiting the past, acknowledging the present, and imagining the future. So the history of anything is really valuable. Individually and personally, we value milestones in our lives, especially in our families. And often we learn a lot from them that would help us navigate our present and our future. And I believe that it should be the same for every discipline that is relevant for us, as we aim to be competent and excellent in a chosen field of profession, and that is, of course, agriculture. And I really like what Robin Sharma had said, that the beautiful value of the past is the platform it presents for even better possibilities. Let me repeat that. The beautiful value of the past is the platform it presents for even better possibilities. Some authors actually have arbitrarily divided the timeline of biotechnology into three: ancient biotechnology, classical biotechnology, and modern biotechnology. And I have added the fourth one as future biotechnology. However, many authors would just list down the major and relevant discoveries or milestones rather than divide the history of biotechnology into certain eras, because it is actually difficult to establish the boundaries for each. But for this slide, we are going to learn a few details on the arbitrary division. So for ancient biotechnology, this began with early civilization, and Homo species and the pre-Homo sapiens, maybe our cousin primates, would be involved in this era, and it would also include the early development of agriculture and food production. Classical biotechnology is characterized by the discovery and practice of fermentation for home use and industrial use. There's also the mass production of food products and other materials to meet demands of an increasing human population. Modern biotechnology is grounded on the advances on genetics, especially assisted by molecular biology. So you have there the advances in genetic engineering, microscopy, biochemistry. I've mentioned molecular biology earlier, and many other relevant sciences. And what I call future biotechnology would be on the many future possibilities and opportunities based on the advances of related disciplines at present. So these would be the arbitrary divisions of the milestones of biotechnology. Please just consider this arbitrary, okay? But let's have some bits of ancient biotechnology, although there are actually no exact or accurate records for this era. How early humans sourced food by foraging for wild plants and hunting for meat is part of this period. It is also characterized by how early humans had initial ability to store food by either sun-drying or freezing, or both, which are actually considered accidental discoveries. This period also includes their early forms of medicine and treatments for illnesses or wounds, and that is by using leaves and barks and roots of plants. And most movies that are based on primitive stories would always show that every tribe would at least have one herbalist or medicine person, either man or woman. In this period, clothing was also discovered using leaves or fur or animal skin. And ancient biotechnology is also characterized by the early stages of domestication. These days, it is common to hear the words traditional biotechnology and modern biotechnology. So it is actually very important that we would be able to identify which is which. And when we go through the listing of some milestones in the history of biotech, we would be able to identify if such discovery is part of traditional or part of the modern biotechnology. But let's go back and learn where the word biotechnology came from. So it comes from two words, biology and technology, which was coined by Karoli Ericki. He was an agricultural engineer from Hungary, and this was recorded in 1919. And for that, he was given the honorary title Father of Biotechnology because he actually published the book Biotechnology of Meat, Fat, and Milk Production in an Agricultural Large-Scale Farm. Of course, this is now the English translation, but it is on record that he was the first to use the word biotechnology. If you'd like to learn more about him, then you can read the article of Fari et al. Published in 2006 that would give more details about engineer Ericki. As I have said earlier, there is really no way that we could have a complete rundown of the history of biotechnology, but what I did was select some milestones and related relevant events for us to go through. And those that are in italics are the relevant events that actually help further enrich the field of biotechnology. So these are not actual biotechnologies, but other discoveries that have helped propel the growth or progress of biotechnology. And the dates are very important, although what I made was select certain events that are actually in huge gaps. So please keep in mind that these are just samples, and there could be a lot of discoveries in between. So let's start with 4000 BC, or around 4000 BC, where Egyptians were able to discover and use yeast to rise their bread and to prepare wine. And in the early years part of this story, we know that Egyptians and other early people would prepare bread that we call unleavened bread, and those are very hard bread. And after this discovery, they have started to prepare what we now call the leavened bread, or the soft bread. And because we have biology, we know that yeast can actually break down sugar to carbon dioxide and water. And it is the carbon dioxide that makes the bread rise and the beverages bubble. So this is actually the beginning of the process of fermentation. So jumping off to around 3000 BC, that's a large leap of around 1000 years. So in South America, in the country of Peru, okay, that is where the center of origin and diversity for potato. Peruvians have selected for larger potato tubers and also frost-resistant tubers, because actually they grow sweet potato and the non-sweet potato varieties up there in the mountains. And Peruvians have also used the different growing techniques and crossbreeding. So this is the early form of crop improvement or plant breeding. Then around in 1763 BC, that's really a huge leap, Sumerians invented the brewing beer, okay, maybe banking on the discovery of the Egyptians early on. And in 1683, so now this is the modern time, at least compared to the BC. So Antoine van Leeuwenhoek, say that again, Antoine van Leeuwenhoek invented the microscope, which allowed scientists to study microorganisms, even molecules, that cannot be seen by the naked eye. So now there is a way to learn and discover about microorganisms, which are very, very important and useful in many byproducts. In 1861, Louis Pasteur developed the process called pasteurization, named after him. And this process actually destroys harmful bacteria in milk and other products. And the process does not affect the taste of food or the nutritional content of the processed foods. And this particular discovery has a huge impact on the food industry. In 1865, Johann Gregor Mendel, who was both monk and scientist, studied the traits of garden pea. He started with 9 and then finalized with 7, and discovered the basic laws or principles of genetics, although his discoveries were largely unacknowledged until these were rediscovered by 3 scientists named De Vries, von Chesermak, and Correns in 1900s. So jumping to 1879, William James Beal developed the world's first hybrid corn by crossing white and yellow corn plants and eventually producing yellowish white corn. And this is now a more structured approach to crop improvement, because James Beal has already an understanding of the basic laws of inheritance and variation from the discovery of Mendel, compared to what the Peruvians had done in the early times, wherein they just sort of select and select. without really understanding the basis for their selection. Now, jumping to 1944, three scientists named Avery, McLeod, and McCarthy established that the DNA and not other macromolecules is actually the hereditary material or the gene. Okay? In 1950, Barbara McClintock discovered jumping genes in her study of Indian maize. So, earlier scientists believed that genes are permanently fixed at their positions, but Barbara McClintock proved them wrong because some genes in the genome of an organism, especially Indian maize, could actually be very mobile. In 1953, James Watson, Francis Crick, Morris Wilkins, and Rosalind Franklin all contributed to the major knowledge that we have now in genetics, and that is the molecular structure of the DNA. Remember that early on, the DNA was identified and validated as the genetic material. So there was now a competition to have an explanation on what is its molecular structure. So now we know that it is a double helix and it can replicate and it has the ability to express its genetic information. So gene expression means that transcription happens and translation happens. So DNA is transcribed into messenger RNA and messenger RNA is translated into protein. And this contribution of the scientists revolutionized many other sciences. So in 1973, Cohen and Boyer show that a gene from one organism can be actually transferred into another organism and the gene remains functional. What we mean by functional is that the gene could produce the protein product that it is coding for. So what they did was take a gene for ribosomal RNA from toad DNA and they cut this using a restriction enzyme or a restriction endonuclease, which has the ability to cut at specific sites in the DNA. So they call this enzyme the molecular scissor. And they inserted this gene into a plasmid. So if you remember your basic genetics, a plasmid is a small circular extrachromosomal DNA which is independent. And they joined the plasmid and the toad DNA using the enzyme ligase. And this combination of plasmid plus toad DNA was introduced into E. coli, and the bacterium was allowed to multiply in culture, and they found out that the toad ribosomal RNA was actually produced in the cells of E. coli. So this was the start of recombinant DNA technology or early genetic engineering. Moving on, in 1982, human insulin produced by E. coli is considered as the first product of modern biotechnology that was released commercially. So this was the product or a product of genetic engineering. So remember that Cohen and Boyer showed how possible it is to do this process, and a company was able to do that. And it was especially momentous because sometime in the 1980s, there was really a rise in the cases of diabetics. So they needed this thing because human insulin was really badly needed. So the E. coli that has the human gene to produce insulin is now called a Just in case you sort of forgot some details about gene expression, I added this slide to refresh your memory with some visuals. So again, transcription of DNA to mRNA, then translation of mRNA to protein would compose the process of gene expression. So when someone does genetic engineering, it must be assured that the gene or genes being transferred from one organism to the next should be able to maintain its integrity to code for the same protein molecule in the recipient organism. I hope that is clear, and if you have some questions, you can clarify with me by commenting on the comment section or in our Google Classroom. To continue, this is your assignment. Why was there a need for insulin to be produced through genetic engineering? So previously, insulin for people with diabetes was produced from where? Okay, this is just a very simple assignment worth 10 points. And for your answers, they should be handwritten in yellow or printer paper with name, subject, and section. Take a photo and email this file to me. You can convert this into PDF file, but make sure that you follow the format for the file name. So the name of the student, your name, and then Agric 27, your section, _assignment, and the date of submission. And this assignment is due on or before September 29. So that is Tuesday next week. And I think it's a very doable and a very interesting assignment for you. So as people being trained in science, I really encourage you to constantly ask yourselves this question as a reminder. What could be the rationale or reasons for these modern biotech or genetically engineered organisms, such as bacteria, plants, animals, and other living organisms, to be desired and developed? Why were they needed? Because there must be some valid reasons or solid reasons for their development. And similarly, we are asked for our rationale whenever we propose a possible research study or any major activity, whether academic or non-academic. To continue, in 1987, DNA fingerprinting was used to convict a crime suspect in the United Kingdom. So like our fingerprints, our DNA profile is actually also very unique. And so this is called our DNA fingerprint. And this is the first time that modern biotechnology was used in forensics. Now in 1990, recombinant chymosin produced in E. coli was released commercially. And this enzyme is actually used to coagulate milk for cheese production. Now, we know that cheese is actually a global food product, so there's a lot of interest to make sure that cheese production becomes more efficient. Now, the original gene that was used to make E. coli a recombinant bacterium was from yeast. Eventually, this product replaced the enzyme that is derived from calves. Anyway, so in 1991, the first transgenic animal was born, and that's Tracy. So Tracy is not very popular compared to Dolly the sheep, but we'll talk about Dolly later on. So it's a sheep with human genes for production of human therapeutic proteins in her milk. In 1994, flavor savored tomato with delayed ripening trait and higher solids was the first genetically modified crop to be commercially released. Now, DRT is a very interesting trait that I would try to elaborate a little bit later on. In 1996, several modern biotech crops, which are either genetically modified or genetically engineered, were introduced into the global market, such as insect-protected corn and cotton, and these are the BT crops, and herbicide-tolerant soybean, corn, rapeseed, and cotton. Now let's have a break of the timeline and look at this photo of an old man who is trying to scavenge some useful tomatoes from a dump of rotting ones. Health-wise, this is really very risky. And I think this picture is from Bangladesh. And in our country, I think there are some occasions wherein we see a similar event. Now, I pulled out this picture and I included this in the lecture today in relation to the delayed ripening technology that was developed in tomato in 1994. So we actually needed this particular new trait because of many related problems. And some of these would be the natural characteristic of some crops. Like tomato, many vegetables are highly perishable and we don't have adequate storage facilities. Many farmers don't have proper storage facilities like refrigeration. And there's also oversupply in the market. Several times this happens, and actually some Benguet farmers had this problem way back in the first quarter of the year. Another concern is the poor farm-to-market road systems until now, and this is 2020 already, and we still have those concerns. And another one is there is less or no access to transportation infrastructure. So some farmers don't have really their own tracking systems, and they're very dependent on renting vehicles and other ways. So, and there could be more than 5 problems related to the need to make sure that many perishable produce from our farms do not perish that easily, because it would be a total loss. So the question is, what can be done? And definitely there had been many solutions in the past, proper packaging from different experts, and what modern biotechnology and what geneticists and plant breeders are offering would be this new trait that maybe we can tweak the genes on delayed ripening, because there are existing crops that would show this natural trait that they don't ripen fast, and that's why you have the ability to preserve the quality of the vegetables or the fruits for a longer time, even if you don't have adequate storage facilities. So these photos would show you the difference between tomatoes with delayed ripening trait and those that don't have the trait. So economically and nutritionally, having this new trait added to many highly perishable vegetables and fruits would actually be a win-win situation for farmers and consumers. The genetically engineered Roundup Ready technology will also be discussed in class, but the details of this discussion will be done in a future lecture, because it is important that we understand the mechanism and process of how such Roundup Ready or glyphosate resistant crops are developed. So plants that die when sprayed with the Roundup herbicide have one step in the shikimate pathway. So I'm showing you that now in this slide. So there's one step in the shikimate pathway that is blocked, and that is the function of the EPSP enzyme. So the 3-enolpyruvyl shikimic acid 5-phosphate synthase, and glyphosate would deactivate or inactivate the function of this enzyme, and that would actually cut off the production of 3 aromatic amino acids that are very essential for protein synthesis. So we talk about tryptophan, phenylalanine, and tyrosine, which are important amino acids for protein synthesis, and when that happens, the plant actually dies. In contrast, the Roundup Ready resistant plants actually have a very functional EPSP synthase enzyme that is not inactivated by glyphosate. So with a functional enzyme, aromatic amino acids will be continuously produced and the plant continues to live. Now, how come that the EPSP enzyme here is still functional? It is because this particular enzyme was coming from or was coded by a gene borrowed from a bacterium. It has a similar function, but it has some unique properties. That's why it is not affected by glyphosate. And so the shikimate pathway is still completed, and the production of the 3 essential aromatic acids continue, giving rise to the continuous photosynthesis and growth and development of plants. But we will discuss this, we will come back to this again in the future lecture. So to continue, in 1997, Dolly the sheep became the first cloned animal. So Tracy was a transgenic sheep, but Dolly is a cloned sheep. and she was developed by Dr. Ian Wilmot and his colleagues at Roslin Institute near Edinburgh. And Dolly actually was cloned from an udder cell of an adult sheep. And of course, several samples of udder cells were obtained and fused with an enucleated egg cell. Enucleated egg cell means the nucleus was removed. And the fused udder egg cell was allowed to divide, that is through mitosis, then implanted into a different sheep, which becomes the surrogate mother. And Dolly was successfully born out of the surrogate mom. So you can see a picture of Dr. Wilmot and Dolly on the left corner. In 2002, the Human Genome Project was successfully done. So they were able, all the scientists and researchers involved in the project successfully sequenced several billion base pairs of the human genome. And this particular milestone would have a very critical implication on human genetics and how diseases that are inherited can be potentially treated. In 2003, the areas planted to GM crops increased by 81 million hectares in 17 countries, indirectly telling us of the interest of many farmers to grow this particular types of crop varieties. Now, in 2006, the first modern biotech drug was approved for release in Europe, then in the USA in 2009, and this was the anti-clotting protein that is produced in goat milk. So imagine if you have problems on clotting. So arteries and veins are blocked because, blood has clotted. And so there should be a way to sort of dilute again the clotted blood in order to have workable treatments done to the patient. So that is the first modern biotech drug that was commercially approved, first in Europe. So keep that in mind. Europe actually has had projects on modern biotechnology and genetic engineering. In 2009, the first blue rose was released commercially in Japan after about 2 decades of research. So that is really a commitment on the part of Japanese researchers to really produce a Japanese or a blue rose. In fact, really many of them are into blue rose and the black rose. There has been a long fascination to produce blue or black rose among many scientists, and finally a research company in Japan named Suntory successfully did it in 2009. So this slide briefly shows you the blue roses developed using different methods. So the one on the left was developed using conventional breeding, and the one on the right was done using genetic engineering, so the modern approach to breeding. And what Suntory actually did in order to produce a bluish looking rose is to borrow at least one gene from this plant. So this is the plant Delphinium, which has very beautiful blue flowers, but maybe not as economically important as roses. So they borrowed one of the genes for blue pigmentation and successfully added that to the genome of a rose variety, which is now able to produce many blue pigments that allow the petals to look more bluish. So that is really very interesting. So when you talk about genetic engineering or breeding, it's not always about increasing the yield of grains and other food crops, but it's also about, making something more beautiful, because a lot of people would have a very good interest or would really value also something that makes them happy and those that really look beautiful for them. So this is actually a highly collectible variety. Moving on. In 2012, Doctors John Gordon of Cambridge University in UK and Shinya Yamanaka of Kyoto University in Japan both won the Nobel Prize in Physiology or Medicine because they discovered that mature specialized cells can be reprogrammed to become immature cells. And these immature cells would be capable of developing into all possible tissues of the body. So think about the many possibilities that using immature cells, damaged organs of people, damaged tissues can be cultured or produced again by a person. And another glaring milestone in 2012 was that there were more than 170 million hectares planted to GM crops in 30 countries. So the last statistic that I gave you in the previous slide was around 17 countries growing this GM crops, and now it has jumped, the number has jumped to 30. And in the country alone, in the Philippines, we have about 800,000 hectares planted to BT corn, RR corn, and the stock traits of BT and RR in some corn varieties. Okay, now in 2013, CRISPR-Cas9 was developed or discovered, and this is actually a gene editing tool. It was discovered in bacteria that operates like an immune system against invading viruses, and many scientists saw more potentials for using this genetic mechanism. So CRISPR actually stands for clustered regularly interspaced short palindromic repeats, and I will not give a lot of details for now, but definitely this would be one that we will come back to in a future lecture. Although I'm going to show you some kind of graphics, I think, in the next slide. Now, in 2015, the vaccine for Ebola was developed by Canada. If you don't know about it, Ebola was actually first discovered in 1976 in Africa, and there was a huge outbreak in the same continent between 2014 and 2016. And at least in between that, a very functional vaccine was developed by some Canadian scientists, and we hope that the same could be done, that in just a few weeks or a few months from now there would be a very functional vaccine against COVID-19. I think everybody, all of us, would wish that and would pray for that. Now, in 2017, China reported to have successfully cloned monkeys, and it is the first time that such technology was done on primates, and just imagine the possibilities if it can also be done on humans. And right, it is still an ethical discussion, but yeah, that is already a matter of interest for now. So I'm showing you briefly the technology called CRISPR-Cas9, which is for gene editing, and as I've said earlier, we will come back to this in a future lecture and try to really understand the mechanism and the process, because this technology is really very powerful and has a lot of potentials and possibilities for future biotechnology across the spectrum of microorganisms, humans, animals, and plants. Okay? So I think we're about to end this lecture, and because we went through the timeline, and that was actually quick if you're going to read a book or several articles on the history of biotechnology, there would be several pages of such topic, and I don't think you will finish reading those materials within the time that we're doing this lecture. So just remember again that I just pulled out very significant events or milestones in the history of biotechnology. But what we would like to emphasize is that for biotech and for all other sciences, we really need to value history. So again, let's have the words of Robin Sharma. The beautiful valley of the past is the platform it presents for even better possibilities. And our field of agriculture definitely needs this boost, this motivation, and this encouragement that if we appreciate the past, the developments in the past, and the problems of the past, it should be able to push us further and farther in terms of innovating things to make agriculture a more powerful discipline and industry. Okay? So, ending with this slide, how about future biotechnologies? And this is a statement coming from one particular journal, and that journal said, specific areas of biotechnology show promise for likely application in 2025, such as personalized medicine, because the human genome is already sequenced, genome engineering, human gene therapy, biomanufacturing, biofuels, environmental remediation, genetic engineering of disease and drought-resistant plants, synthetic biology, and technologies at the intersection of nanotechnology, biotechnology, and engineering. So, again, if we just consider and have a mindset that is more positive on this technology and many other technologies offered to agriculture, then we can really push forward our vision of a well-developed agricultural sector in the Philippines. And with that, I'm going to end this lecture, and I'm going to see you in our Google Meet schedules. And if you have questions or clarification, make use to raise them during our Google Meet, or you can already post them in our Google Classroom so that I could also be made aware in advance and try to answer that during our Google Meet. And I would leave you some of the beautiful words of Mr. Lang, and that is, technology is a useful servant but a dangerous master. And I think what he meant by this is that biotechnology and many other technologies are actually very useful and powerful, but we as users or developers of such technologies should be very, very responsible. Okay? That is really a key word, responsibility, being very responsible, because such good and useful technologies may be misused. And yep, those would be some thoughts for you to ponder on while you wait for our Google class. And with that, I say bye and have a wonderful day, everyone.